In this video, we're going to take a look at the concepts known as osmosis and dialysis. These are very closely tied into concentration of solutions, as we'll see. The key here, and where it becomes important for us biologically and from a health perspective perhaps, is that cell membranes, remember back to some of your basic high school biology days, cell membranes are selectively permeable. And so this means that membranes are going to allow certain things in and out, but only on a selective basis. And some of that transfer across those membranes is going to be determined by some of these aspects of osmosis and dialysis that we talk about. And so, as I said, the selectively permeable membrane means that only certain substances are allowed to pass across the membrane, and that cell membranes will control what actually enters and leaves the cell. Some of the most important factors in determining whether a solute can cross or not are the size of the molecule and the polarity of the molecule. And so we can see here that very small nonpolar molecules, things like O2, CO2, and N2, they're very permeable to a membrane. They'll pass through almost unimpeded. When we start to get to small but uncharged polar molecules, things like water or glycerol, a little bit easier for those to go through. Things like glucose and amino acids, very large, uncharged polar molecules, or ions, or large, very touchy mouse for this lecture, or some of the larger things like proteins and sugars, triglycerides. Those have a lot of trouble getting through a membrane. And again, we're going to look at a lot of these factors. The different concentrations of ions on either side of a membrane can be described by these following terms. So hypertonic means that the solution, that the solute is at a higher concentration than the solution. So again, a hypertonic means that our solution on one side of the membrane is at a higher concentration. Hypotonic, our solution we can consider it to be maybe outside of the cell. Hypotonic means that our solution outside of the cell is at a lower concentration, whereas isotonic means that we have equal concentrations on both sides of our membrane. Ultimately, as long as the materials that we're talking about can pass through the membrane, what is going to occur is ideally the solutions on either side of the membrane coming to the same concentration. And that happens through different means, again, depending on what can actually pass through the membrane. Osmosis refers to the flow of the actual solvent. And so the solutes don't move in the case of osmosis, but the solvent does, typically water. And we'll look at some examples of that. But that would mean, for instance, if you have a higher concentration on one side of the membrane, then water is actually going to flow across the membrane until those concentrations are equal to each other. Not the particle itself, but the water will flow with osmosis, which is the opposite of dialysis. And dialysis, now the water is going to stay where it is, but the actual solutes are going to move across the membrane. Solute particles are always in constant motion, called diffusion, just diffusing through an area. Solutions that have different concentrations create what we call a concentration gradient. And so if you have a different concentration on one side of the membrane as opposed to the other, then you have a concentration gradient across that membrane. And either osmosis or dialysis may occur, again, depending on what type of membrane it is and what can actually pass. But either osmosis or dialysis could occur in order to equalize that concentration gradient. In the case of dialysis, solute particles will diffuse from hypertonic, the area of higher concentration, to hypotonic, the area of lesser concentration. And so we can see an example here. And so this example is looking at osmosis. When the solutes cannot pass through the membrane, then the solvent moves instead. In the case of osmosis, it's the opposite. The water is going to move from the hypotonic to the hyper. And that should make sense, right? Because if we have, so in this example here, we have this membrane that is only permeable to water. And so we have a hypotonic situation over here, hypertonic over here, higher concentration. So we have a concentration gradient between the two. And so the water is going to flow in this direction. The water is going to flow from solution A to solution B. It's going to move from the hypotonic to the hypertonic, because as it does so, as water leaves the solution, we haven't changed the amount of solute particles, but our volume is changing. That means our concentration of solution A is going to increase. Because if we remember what concentration is, so concentration, if we're thinking in terms of molarity here, concentration is our number of moles divided by liters. And so if we decrease, actually, let me just, do it this way. So that's going to equal our molarity. So if I decrease my volume, if I decrease that number of liters, that's going to increase the molarity. And so then water is moving, oops, sorry about that. Water is moving into our hypertonic chamber. And so our volume is increasing there, which will lead to a decrease in the molarity. And so that will happen until the molarities on both sides are the same and we no longer have that concentration gradient. So that's osmosis. The water moves across. The hypertonic solution in a case of osmosis will always increase in volume, again, in order to equalize that concentration gradient. What we call osmolarity is determined by the total concentration of the solutes. The identity doesn't matter, the size doesn't matter. We're looking at the concentration of the solutes, which creates that concentration gradient. Osmosis can be stopped or even reversed by applying some sort of external pressure. So you might have heard, for instance, of a reverse osmosis water filtration system. Goes in the exact opposite direction, but some sort of pressure has to be applied in order for that to occur. The osmotic pressure is the amount of pressure needed to stop that flow. And so like the example that I just used, reverse osmosis, used to purify seawater, for instance. It's also used to remove any excess water in hemodialysis treatments, which some of you may be familiar with. Then we see just another example here, seawater and freshwater. So we have our selectively permeable membrane that is only going to allow water across. And we can see here that water is going to move into the chamber that has seawater in order to equalize these concentrations. So here's our hypertonic side, our hypotonic. So water will flow over until that concentration gradient is equalized. The osmotic pressure in a reverse osmosis system here, that's going to be the amount of pressure that's applied to prevent that from happening. So again, normally water would be flowing in this direction in order to equalize this gradient. But what we're going to do here now is apply this pressure down onto the saltwater chamber, which would not allow that to happen. And so now instead, water is going to flow. So now instead, our water is going to flow in this other direction. And that's one of the ways that we can desalinate the water. So we set up this chamber, which now has an osmotic pressure. So the water wants to flow, but we apply pressure to reverse the direction of that flow. And that's going to then take some of the water that was here, basically pump it over into our freshwater side, but it's only going to move the water. And so what'll be left over here is just a little bit of water and a whole lot of salt. And so again, just some of the terms that we use, the hypertonic solution is our more concentrated solution. Hypotonic, the more dilute. Isotonic, solutions with equal concentration. We can use this to predict our direction of osmosis. The osmolarity is our total concentration of all of the solutes in the solution. here's a look at where it might happen with red blood cells. And so this is one of the reasons why it is important to give electrolytes to patients possibly. And so here's a red blood cell in solution. If you have a completely isotonic solution, we have the same amount of solute particles inside and outside, so everything's fine. If, however, you drop that red blood cell into a hypotonic solution, so remember, water is always going to move. Let's come back and take a look at our, where'd it go? So osmosis always occurs from the hypo into a hypertonic. So now our solution is hypo, our cell is hyper. And so water then, in this case, so if we drop a red blood cell into a hypotonic solution, water is going to move into the red blood cell, possibly to the point where it's going to burst. If we drop it into our red blood cell into a hypertonic solution, now water is going to move from the cell into the solution, causes it to shrivel up. And so we need to make sure, that's one of the reasons why it's important to keep a balance of electrolytes in our system so we can avoid the sort of problem with our cells. So here we just have an example of aqueous solutions A and B, separated by our selectively permeable membrane. Again, it's the same sort of idea. Osmosis will occur in order to increase our concentration on this side and decrease our concentration over here until A and B are equal. Dialysis is the diffusion now of solute particles across a membrane. So in dialysis, the water molecules aren't moving the way they do in osmosis. With dialysis, the actual solute particles are going to move, and they're going to move from a hyper to a hypotonic solution. And so here we have an example. And so in this example, we have a mixture. We have our selectively permeable membrane, so a dialysis membrane. This is quite often done, maybe if you're working in a lab, you might fill, I used to have to do this for certain experiments that I did. I would fill a dialysis membrane, some dialysis tubing with a mixture, and then I would leave it in water and spinning and circulating overnight, because then the things that I did not want anymore in my mixture would separate out. And so that's what's happening here. In this case, what's happening is that our membrane will only allow these smaller blue particles to come through. And so mixture A contains urea, which are these smaller blue circles, and albumin in yellow. And so what's going to happen is that the urea is going to pass through the membrane, but the albumin can't. And so over time, that albumin is going to stay where it is, but the urea is going to pass across the membrane. In the experiments that I used to do, I used to have to prepare a protein, and I'd have to make sure that all the metals were removed from my mixture for various reasons, for things that we ended up doing, experiments we ended up doing. I had to remove any trace of any metals in there. So I used a compound called EDTA, which will go around and basically scarf up and grab all of our metal ions. But then I needed to remove all of that from my solution. And so I would put my whole mixture of my purified protein with my EDTA, I would put that into a dialysis bag, and then I would set it into a huge beaker of water. And then I would just allow the whole thing to spin for a while and sit in that water. And what would happen is that the EDTA would be allowed to pass through the membrane, but the protein that I purified was too big and it couldn't. So the EDTA passes out, and eventually I'm left with just my purified protein inside my dialysis tubing. So it's a great way to separate things, as long as the sizes of your particles are such that you can do that. And so back to our red blood cells, and I'm sorry these are a little bit out of order, but this is exactly why IV solutions must be isotonic. If IV solutions are not isotonic, then red blood cells are affected. And so that is our presentation on osmosis and dialysis. Thanks for watching, and you stay classy, USM.